everyone. My name is, and yeah, I'm a currently third year PhD student. And today I'm going to introduce our recent work, the scReadSim. This is a single-cell RNA-seq and ATAC-seq read simulator. And so I want to start with this figure. This is a review paper published in Guanao Biology last year. So it summarizes the analysis tools developed for just the single-cell RNA-seq data. So you can see there has been over 1,000 tools developed for just single-cell RNA-seq. So for the users or the researchers, if you want to make decisions, which analysis tool is better for us to use, it could be tricky. So one solution could be we use some synthetic data with ground truth. So we use some simulator to model the real data to generate the synthetic data, and we have the ground truth. We use that data to benchmark these analysis tools. Then we can find which one is better for you for the specific target you're working on. But let's have a closer look into those analysis tools. So in our discussion, we separated the analysis tools into two parts. So the first category is the low-level analysis tools. The other one is the count-level analysis tools. So for count-level analysis tools, basically, these analysis tools can only work on the count matrices. So that means we need to first count the sequencing reads from the BAM file or FASTQ file, like count how many reads have been overlapping with each feature for every single cell. And we use that processed matrix as the input for the count-level analysis tools. But for the low-level analysis tools, these tools can directly work with the sequencing reads. So in this case, if we so another fact is for all these count-level analysis tools, the most existing simulators we have are the count simulators. So it's like most of the existing simulators, they can only generate the count matrices. That means we cannot use those count simulator to help benchmark the low-level analysis tools that take reads. So that means we need some simulator can directly model the sequencing reads and generate the synthetic sequencing reads, have the ground truth for us to benchmark the low-level analysis tools. So that's why we propose this scReadSim. This is a single-cell RNA-seq and ATAC-seq read simulator. So there are actually existing one or two read simulator for the single-cell RNA-seq or single-cell ATAC-seq, but those methods are not trying to mimic in the real data. So their target is like providing a lot of free parameters for the users so they can arbitrarily design their single-cell RNA-seq data or single-cell ATAC-seq data. That means their synthetic data is not necessarily mimicking the real data. So it might be not trustworthy. But our target here is first, we want to keep the ground truth. We can let the users have the ground truth to benchmark the tools. That's the simulator's purpose, right? Secondly, we want to provide some synthetic data that can preserve the real data's information, can mimic the real data. So that's our advantage here. And in addition to that, we can also have the flexibility, like provide some parameters that the users arbitrarily design their data. Like we can let the user to vary the cell number or the sequence depth. And for single-cell ATAC-seq data, we can let them even arbitrarily design their open chromatin region. So the peaks, they can specify that for their synthetic single-cell ATAC-seq data. And here is a general workflow for the scReadSim. So basically, we need users to input first the real BAM file that contains all the sequencing reads for their single cells, and also the reference genome. That is the genome that all the single-cell sequencing reads have been aligned onto. And for single-cell RNA-seq, we need the users to specify the gene annotation file, the GTF file, which could be a public resources, accessible resources. And for single-cell ATAC-seq data, we need the users to input the peaks. So this could be the open chromatin regions identified using any peak calling methods. So here, the reason we want to leave this choice to the users is the most important thing is, so for the real data, the single-cell ATAC-seq data, no one knows the ground truth. We don't know where the open chromatin region are. So we need to use peak calling methods. And the other thing is, there are a lot of peak calling methods, and different peak calling methods have different performance. And for users, they may have different tastes of which one is better. They prefer which one, I don't know. So we want to leave this choice for the users. And given those input files, like the genes, gene annotation files, and the peaks, we construct the foreground features and background features. So here the foreground features are genes when analyzing single-cell RNA-seq data, are peaks for single-cell ATAC-seq data. And the background features are the intergene region or the interpeak region. So if we look into this reference genome, if the user has specified this part to be the gene or to be the peak, and then we take the remaining reference genome area as the background region. So in this way, we separate the whole reference genome into two categories of features, foreground feature and background feature. And then for foreground feature, we count this read count matrix. For background feature, we also do this step to construct this count matrix. And then we train our previously designed read count simulator, scDesign2, onto this read count matrix to generate a synthetic count matrix. So the reason why we chose scDesign2 is our simulator, our read count simulator can well preserve the joint distribution of the read count across all the genes or all the peaks. So that means in addition to the marginal distribution of each read count for every gene or peak, we can also preserve the gene-gene correlation or the peak-peak correlation. So in this step, we can make sure that our simulator, under the read count level, we can preserve all the information or the correlations among the features in the real data. And given these synthetic count matrices, we can use this number to indicate how many synthetic reads we need to generate for every synthetic cell within each feature. To do so, we first need to pull all these sequencing reads from the real BAM file into these pseudo bulk reads. And then we will sample the corresponding number starting positions from the real reads for every synthetic cell within each peak. And then we will extract the corresponding sequencing information from the reference genome after the user has specified the read length. And also we will preserve the substitution narrow and the chemospectrum, all these sequencing characteristics from the real data to generate the synthetic reads. And after that, we will have the synthetic reads for each synthetic cell across all the regions, all the features, like the foreground feature, background feature. And then we put them together as an output in FastQ or BAM format. And those are the standard input for the low-level analysis tools. So this is a general workflow for the scReadSim, and I will go into detailed version for RNA-seq and ATAC-seq later. So first, for single-cell ATAC-seq data, here is how we deploy the scReadSim onto the single-cell ATAC-seq data. So as I mentioned, we need users to input the peaks for the single-cell ATAC-seq data. So here, this is the genome browser view of two BAM files. So this track is the real BAM file. Those are the real data. And this one is the synthetic data, the BAM file we generated here. And those dark blue bars are the real peaks input by the users. And here we use this max3 to call peaks from the real data and input into our simulator. So you can see here, given those ground truth peaks, we can let our synthetic data preserve the read summits across those peak region. And also for the non-peak region, the read coverage can be distinguished from the ground truth peak region. So that means we can preserve the real data's read coverage into our synthetic data. So we have the ground truth here, the peak and non-peaks position. And here are some quantitative metrics that we used to justify that our data are really mimicking the real data. So this is from the read level. Like for the B panel, we examined the fragment length distribution for the real and synthetic reads. Because for ATAC-seq data, we know that they are a paired. That means we have some distance between the two endings of the read pair. And that distance is called the fragment length. So we draw that fragment length distribution for all the real reads and all the synthetic reads. And you can see the distribution from the real and the synthetic data are pretty similar. And C panel is we examine the substitution narrow of each base position in the read. And you can see these two curves are basically overlapping with each other. So our synthetic data can preserve the substitution narrows from the real data, real reads. And D panel is we use some normalized read density to examine the read coverage for every feature. So here we calculate this RPKM, the read per kilobase milling. So basically we just count how many reads have been overlapping with each feature, and then we divide that number by the length of that feature. And we summarize these quantities, these statistics for the peaks and non-peaks separately, and we look into this distribution. So here you can see for the peaks, the read densities are higher than the non-peaks. Similarly, we can find this distinction in our synthetic data, and the distribution quantities, the statistics are basically similar between the real and the synthetic data. So these are some part of results we used to verify that our synthetic data is mimicking the real data. And this is from the read level. We also did more verification, like using the, we verify it under the read count level, like by examining this step. After the assay design to generate the synthetic count matrices, we verify that this count matrix is mimicking the real count matrix pretty well, like by preserving the correlation among the features. Because this is a pre-step for our read generation, and it's quite important to make sure all this information from real data is well preserved. And in addition to that, so when we deploy the scReadSim onto the single-cell ATAC-seq data, we can make sure that our synthetic data have the same open chromatin region as the real data. But we can do more. Here, we can let the users to arbitrarily specify their synthetic data's open chromatin region. So that means, in addition, so that means the user first need to specify the trustworthy peaks for the real data. That's this track here. And besides, they can also specify any arbitrary regions they want to have on the reference genome to let their synthetic single-cell ATAC-seq have as the open chromatin region. So here, this track is the real peaks, the trustworthy peaks, the input for the real data. And these dark blue bars are any, like we randomly generate those regions as the target synthetic peaks for our synthetic data. So you can see here, the synthetic data, they can preserve the read summits across these target peaks and can also be distinguished from the non-peak region as specified by the users. And this is a workflow for how we did this part. So there are a lot of technical details. I won't go through them, but the general idea here is the user need to specify the input peaks. Those are the real peaks for the real BAM file. They also need to specify the output peaks. Those are the peaks they want their synthetic data to have. So basically 2 sets of peaks. And then we define some mapping between the input peaks and output peaks. And we construct a count matrix for the input peak, just like what we do in the general workflow. And then we use this mapping to extract the corresponding count vector from the input peak count matrix into our output peak count matrix. And then we follow the same procedure to train our count simulator and then to extract the sequence information to generate the reads for the synthetic reads. And here is how we define the mapping. So given the input peaks, so these are the real peaks for the real data. And here is the output peaks for the user want their synthetic to have. And we define this mapping based on how these peaks length are similar between the input peak and output peak. So here you can see the output peak 1 has the most similar peak length with input peak 2. So we define this mapping from IP2 to OP1. And that's how we define all these mappings for all the output peaks. And then we extract those count vectors for the IP2 to OP1 because we have this mapping here. So that's how we construct the count matrix for the output peaks. And so we can let the user to arbitrarily design their synthetic single-cell ATAC-seq data. We know which part of the region are the ground truth open chromatin region in our synthetic data. Then we can use that to benchmark the peer calling methods. Here is a result that we show our method can be applied for benchmarking the single-cell ATAC-seq peer calling methods. And the ground truth are the user-specified open chromatin regions, like the output peak I mentioned the term. And here we deploy 4 of the most popular peer calling methods for single-cell ATAC-seq data. Max, the third version of Max, and Seeker, Homer, and HMR ATAC. So these 4 methods, and we obtain the 4 sets of peak calling results from these 4 peak callers. and we compare it with the ground truth peak sets. And here we use this read density to calculate how the read coverage change over the features for the different peak column results and also the ground truth. So we can see here, the max has the most similar results to the ground truth. And also we change the threshold value for those peak column methods to change the number of their discoveries and compare how the discoveries are overlapping with the ground truth peaks. And we draw this TPR versus FPR and precision versus recall curve. And still the max is performing the best. And also the accuracy, regarding the accuracy, the HMMR attack has the similar performance with the max, because the HMMR attack itself is a peak column method developed for ATAC-seq. This is the only one peak column method developed for ATAC-seq data. And here is our application for the single-cell RNA-seq data. So this is the workflow for single-cell RNA-seq data. It's a little bit different from the single-cell ATAC-seq data. So here we need the users to specify the gene annotations. So we need to know the locations of the genes. And here we do not have the arguments about the peaks, the ground truth peaks or the non-peaks, because we already know which region of the reference genome are the genes or non-genes. So we don't need to worry about that. So the ground truth here in our simulator is trying to provide the UMI count for the UMI-based single-cell RNA-seq data. That's the ground truth we want to provide here. And going back to the workflow, after the users have specified the genes, we will construct the read UMI count matrix for the genes, and also the read UMI count matrix for the intergenes. So these are the background regions. And then we still train the assay design tool simulator to obtain the synthetic count matrix, synthetic UMI count matrix. And also for the background region, we generate this synthetic UMI count matrix. And these count values are indicating how many unique molecules we need to have for each synthetic cell across each feature. So from this result, from this number, we only know how many unique molecules. We do not know how many reads there are in the synthetic data. So we still need to model that from the real data. To do that, we first pull all the reads from the single-cell RNA-seq data, the real data. And we construct this empirical distribution by counting how many reads have been assigned for each UMI. And we use that empirical distribution to sample the number of reads for every UMI in our synthetic data. So basically here, after we got this count value, we generate like n UMI strings by randomly permutating the ACGT strings. And then for each UMI, we sample the number of reads from this empirical distribution. And then we generate the corresponding number of synthetic reads for that UMI. And then we assign all these reads with the same UMI, and of course the same cell barcode, because they're from the same cells. So that's how we generate the synthetic reads for the single-cell RNA-seq data. And here, this synthetic count matrix can be taken as the ground truth of how many unique molecules we have in our synthetic data. And this can be used to benchmark the single-cell RNA-seq analysis pipelines or the deduplication methods. So the deduplication methods, they are like taking into the BAM files, the reads, the single-cell RNA-seq reads, and they count how many unique molecules for each cell within each gene, and they output a UMI count matrix. And our simulator could provide the ground truth of the UMI count that can be helped benchmark those tools. And here is the result we compare. We benchmark two of the most popular single-cell RNA-seq deduplication methods, the cell ranger and UMI tools. So the first benchmark result is for the correlation. So after we deploy these two deduplication methods, we obtain two UMI count matrix, and we compare it with the ground truth UMI count matrix. So the first thing is we calculate the correlation between the two count matrices. And this rule is for the gene-wise. So basically for every gene, we calculate the correlation between the ground truth UMI and the UMI provided by those analysis tools. And we calculate for every gene and summarize the distribution here. And this is for cell-wise. So for every cell, we do the same thing, and we pull all the cells statistics together to draw this distribution. So the information here you can see is the cell ranger has a better performance than the UMI tools. And we also, I will talk later. So we also can let the user to vary the cell number and the sequence depth. So in that we can, so with that we can benchmark the computational time of each method. So we know those low-level analysis tools, they're quite slow because they need to deal with the raw data, right? So we really want to learn about how they will take to run a whole session for all these reads. So we can see here, if we change the cell number but fix the sequence depth, the elapsed time won't change much for both methods. But if we increase the sequence depth, the library size for the single-cell RNA-seq data, we can see a linearly increasing trend of the elapsed time for those methods. And finally, we compute some statistics of the UMI count metrics and compare it with the real UMI metrics. And still the information is the cell ranger is the top performant method. And we also examine the lower dimensional embeddings of this method. So this is the UMAP of the ground truth UMI metrics, and these two are the UMI metrics obtained from cell ranger and UMI tools. And we show the Pearson correlation, the global Pearson correlation of the ground truth and those UMI metrics. So still the cell ranger is performing the best. And this is a figure to visualize that our simulator for single-cell RNA-seq data, we can let the users to arbitrarily specify their cell number and the sequence depth. So you can see the increasing tendency if we increase the sequencing depth for the synthetic data. And that's basically my presentation today. Here are the materials and the software. Any questions? Go ahead, Matt.
That was just an applause. Sorry, I don't have a question.
Go ahead.
That was just an applause. I was saying, good job.
Okay.
It wasn't a raised hand, sorry.
Thank you so much. Hi.
Question. Great talk. That was really cool and very useful. So I was wondering, because you have a single-cell ATAC simulator and an RNA-seq simulator, but nowadays the measurements you can measure both together.
Yeah.
Could this be extended?
Yes. We actually we have the results like do the single-cell multi-omics data simulator. Yeah, we can definitely do that. Oh, that's a good point. Yeah. Thank you. Go ahead. Okay. The workflow. Okay. You mean for this part? Okay. So basically the input peaks are the trustworthy peaks the user specified for the real data. And the output peaks are the peaks they won't have in their synthetic single-cell ATAC-seq data. And given those two peak sets, we define a mapping between the peaks because we need to extract the real data's information and want to put it into the target peaks for the users. And that's the mapping works. And after we obtain the mapping, we can just like extract the corresponding read count from the input peak to the output peak. And regarding the read position, we can do the shift because we still need to sample the read starting positions from the input peak, and we make a shift towards the output peak, and that's the synthetic read starting position within the output peaks. And regarding the sequence information, that's the same with the other workflow for single-cell ATAC-seq data.
State with the interpreter, and what is the state for what totally different condition? Can you just specify what output peaks you want to see in that different condition, and then use your one way of interpreting that?
Yeah, I don't quite sure. I'm not quite sure the condition means, but if it's just the positions of the peaks, we can definitely do that. Yeah.